Biochemistry 401 Lecture 33. Today we're going to talk about nucleotide function, structure, and synthesis. We're going to talk about the de novo pathway and the salvage pathway. So let's get started. Let's begin by talking about nucleotide function. Nucleotides are precursors for nucleic acid synthesis, DNA and RNA. And this is what we primarily think about when we think about nucleotides. But they're also used for energy sources. ATP is a prime energy source for biological processes, such as molecular motors and active transport of solutes across membranes. GTP is also sometimes used as an energy source. Nucleotides can also activate biomolecules for synthetic reactions, and we've seen this previously. For instance, UTP activating glucose and CTP activating alcohol head groups. And we've also seen ATP activating many biomolecules. Nucleotides and their derivatives are also important parts of many second messenger systems, such as cyclic AMP and cyclic GMP. Finally, ATP is a source of phosphate for kinase reactions, such as the ones that are involved in regulation. We saw this in glycogen synthesis and degradation, and also in glycolysis. So now let's talk about structure. We're going to talk about the structure of nucleosides first. Nucleosides are comprised of two things, sugars and nitrogenous bases. Ribonucleosides are comprised of a ribose sugar and a base, and deoxyribonucleosides are comprised of a deoxyribose sugar and a base. And the difference is shown here. We have adenosine that's shown in the top figure and deoxyadenosine that's shown in the bottom figure. The difference between the two is that deoxyadenosine is reduced at the 2' position, and the arrow is pointing to this position. Now the bases can either be purines or pyrimidines, and we'll see that in just a minute. Nucleotides are nucleosides plus 1, 2, or 3 phosphates. In the top position, we see adenosine, a nucleoside, and in the bottom position, we see adenosine monophosphate, AMP. This is also known as adenylate. In this slide, we see the nomenclature of bases, nucleosides, and nucleotides. It's important that you know the name of the base, the name of the ribonucleoside or deoxyribonucleoside, and the name of the ribonucleotide or deoxyribonucleotide. And so it's important that you know the difference between adenosine and adenylate, for instance. Nitrogenous bases fit into two major categories. Purines consist of adenine and guanine, and if you'll notice, these are structures made up of 2 rings. Pyrimidines have 1 ring, and there are 3 of them, cytosine, uracil, and thymine. To remember this, here are some mnemonics. Pure as gold for purines, A and G. Cut pi, CUT are all pyrimidines. I hope this helps. So now let's look at nucleotide synthesis. There are 2 major pathways. The first is a salvage pathway, in which we use recycled bases and attach them to an activated ribose platform, PRPP, which is 5-phosphoribosal 1-pyrophosphate. The other way is the de novo pathway, and this is making bases from scratch. We start with an activated ribose again, PRPP, and we add amino acids, ATP, carbon dioxide, and other things to make the finished nucleotide. So in the salvage pathway, 5-phosphoribosal 1-pyrophosphate plus guanine, for instance, yields guanosine monophosphate and pyrophosphate, and so this is driven forward by pyrophosphate release and subsequent hydrolysis to 2 molecules of inorganic phosphate. The de novo pathway is a little more complex, and pyrimidines and purines use 2 different strategies. Pyrimidine rings are simple. They're formed first, and they're brought to the activated ribose platform, PRPP. And the subsequent progression, involving several steps, is first to make UTP, and then to make CTP. These are both ribonucleotides that can be used to make RNA. Then UTP is used to make TMP, and CTP is used to make DCTP. These are both deoxyribonucleotides, and so what we do first is we make the base first, we attach it to PRPP, and then we make ribonucleotides. We use the ribonucleotides as substrates to make deoxyribonucleotides. Now, purines are much more complex. They involve two rings. These are made directly on the PRPP platform. First to make the intermediate IMP, then to make ATP and GTP, so we're making ribonucleotides first, and then to DATP and DGTP to make deoxyribonucleotides. Purine rings are formed piece by piece right on the activated ribose sugar. We're going to go over the pyrimidine de novo pathway first as an overview, and then we're going to go through it bit by bit. We're going to do this with the pyrimidine de novo pathway, but we're not going to do the same thing with the purine de novo pathway. So let's get started with this. First of all, we're going to synthesize an intermediate called orotate. This is comprised of carbamoyl phosphate and aspartate. We're then going to form orotadilate. This occurs by attaching orotate to PRPP. Then we're going to form UMP, uridylate. This happens through a decarboxylation of orotadilate. We're then going to phosphorylate UMP to make UTP. And then we're going to aminate UTP to make CTP. And these are all ribonucleotides. The next thing that's going to happen, we're going to make TPP and DCTP. We're going to start with the orotate synthesis. So the first thing that we're going to do is we're going to make orotate. And it looks like this. The green portions come from aspartate, and the blue and black portions come from carbamoyl phosphate. Now that's a name that should seem familiar to you. This is the synthesis of carbamoyl phosphate. The process is essentially the same as what we saw when we looked at the urea cycle. Carbamoyl phosphate synthetase 2 actually catalyzes the rate-limiting step in pyrimidine de novo synthesis, and so it's really important that you understand this process. This is a cytosolic enzyme that synthesizes carbamoyl phosphate. Do you remember where the other carbamoyl phosphate synthetase was? The one that's involved in urea synthesis is actually in the mitochondrion. This one that's involved in nucleotide synthesis is found in the cytosol. And so, just like before, we're going to start with bicarbonate. We're going to phosphorylate it to make carboxyphosphate. We're then going to aminate it to form carbamic acid, then another phosphorylation to yield carbamoyl phosphate. And so this is an activated intermediate, and this is where the difference comes in. If you remember, in urea synthesis, the amino group came from ammonium. In this case, however, the amino group can either come from ammonium or it can come from glutamine. We're going to see that glutamine is often a donor of amino groups. And so what we have is glutamine plus carbon dioxide plus 2 ATPs plus water yields carbamoyl phosphate plus glutamate plus 2 ADP plus inorganic phosphate. And the water that you see there is used in the hydrolysis of glutamine to make glutamate. Now if you look at the enzyme here, it looks similar to the one that we saw in the urea cycle, but there's a glutamine hydrolysis site at the top, there's a bicarbonate phosphorylation site just below it, and then there's a carbamic acid phosphorylation site below that. And so again, this enzyme works by channeling, and this diminishes the loss of substrate by diffusion, and it also helps to prevent non-productive reactions, such as the hydrolysis of ATP, which would be a waste. And so in the end, we have carbamoyl phosphate. This combines with aspartate, and inorganic phosphate is lost. We end up with carbamoyl aspartate. And then through hydrolysis, we end up with dihydroorotate. And then there's an oxidation to make orotate. And so we're going to add orotate to PRPP to make orotadilate. It is going to be added to 5-ribosyl 1-pyrophosphate PRPP through the formation of a glycosidic bond with the loss of pyrophosphate, which acts as a leaving group. This reaction is driven forward through the subsequent hydrolysis of pyrophosphate to yield 2 molecules of inorganic phosphate. The enzyme that catalyzes this reaction is called pyrimidine phosphoribosyl transferase. And this name makes sense. We're transferring a pyrimidine ring to a phosphoribosyl platform to yield a rotadilate. The next step is the decarboxylation of a rotadilate to yield uridylate, UMP. This is accomplished through the loss of the carboxylic acid group that was originally part of the aspartate R group. And finally, again, we end up with carbon dioxide and uridylate. We're almost there. We have UMP. Now, to go from uridylate to UTP, there are 2 kinase reactions that occur to go from UMP to UDP to UTP. And there are specific kinases that do that, and we won't go into this in too much detail. But the next thing that happens is we're going to add an amino group from glutamine, which is a really good amino group donor. And so first we're going to phosphorylate, and then we're going to hydrolyze the amino group from glutamine and transfer it to UTP to make CTP. And so the process of going from UTP to CTP is an amination. Control of pyrimidine synthesis occurs through the regulation of 2 enzymes primarily. Those are carbonyl phosphate synthetase and aspartate transcarbamylase. We're going to look at each of these individually. So let's start with carbonyl phosphate synthetase. This enzyme functionality catalyzes the rate-limiting step of pyrimidine synthesis, and so is very important. This functionality is inhibited by UTP. Now this is the product of the pathway, and so this is product inhibition. It's also known as negative feedback. This enzyme functionality is activated by PRPP, which is one of the substrates in UMP synthesis. Aspartate transcarbamylase is negatively regulated by its product, CTP, and it is positively regulated by ATP, which is a purine. Now this makes sense. CTP will negatively feed back on this pathway to ensure that too many pyrimidines are not made, whereas ATP will increase the activity of aspartate transcarbamylase to ensure that when there are too many purines around and not enough pyrimidines, that more pyrimidines are made. It's important to remember that a stable base pair requires both pyrimidines and purines, and so therefore we must have relatively equal amounts of pyrimidines and purines to make RNA or to make DNA. We're now going to talk about the de novo synthesis of purines. This is a more complex synthetic pathway, and so we're just going to hit the high points. So let's talk about the big picture first. We're going to make an intermediate inosine monophosphate, IMP. This is going to be used to further make ATP and GTP, which can be used for RNA, and then these can be reduced to make DATP and DGTP. This synthesis begins with phosphoribosylamine synthesis. This reaction is catalyzed by the enzyme glutamine phosphoribosyl aminotransferase. This represents the first committed step in the de novo synthesis of purines. The amino group is going to be hydrolyzed from glutamine, and then the ammonia is going to be added to PRPP with the loss of pyrophosphate that is then hydrolyzed to 2 molecules of inorganic phosphate. And so we're left with 5-ribosyl 1-amine and 2 molecules of inorganic phosphate. This is the first committed step, and therefore it is highly regulated. This reaction, like many of the reactions in nucleotide synthesis, is regulated by downstream products. This enzyme is inhibited by AMP and ADP and by GMP and GDP. And here are the next steps. You'll be happy to know that you do not need to know these steps at all. In fact, we're going to go straight from phosphoribosylamine all the way to the next major intermediate. And this is inosonate. This is a common intermediate in both the synthesis of AMP and GMP. Inosonate is also called inosine monophosphate IMP. IMP has a hypoxanthine base. Now the difference between AMP and IMP and the difference between GMP and IMP is the presence of an additional amino group. In the synthesis of AMP, we're going to replace the ketone with an amino group. In the synthesis of GMP, we're going to add another ketone and then aminate that to make GMP. In each case, we're going to phosphorylate to activate the molecule so that we can add an amino group. Let's look at this a little more closely. The synthesis of AMP and GMP occur through a branched synthesis from IMP. One branch gives rise to AMP and the other one gives rise to GMP. And so inosine monophosphate is a common intermediate in the formation of both of these nucleotides. We're going to talk about the synthesis of AMP first. We're going to start with IMP, and then we're going to phosphorylate and add an amino group from aspartate. Now we have a little bit extra that's included in the intermediate adenylosuccinate, and we're going to release that little bit as fumarate, and we end up with adenylate, AMP. I want to draw your attention to the fact that we're using GTP as a phosphate source in the synthesis of AMP, and the reason for this will become clear in just a minute. Now in the other branch, in the synthesis of GMP from IMP, we're going to make the extra ketone first to make xanthylate. We're then going to phosphorylate xanthylate, and then we're going to hydrolyze an amino group from glutamine to make glutamate and free ammonia. The ammonia is going to be added to yield GMP. We're using ATP in this phosphorylation. And so for these syntheses, you need to know what the common intermediate is, you need to know what the final products are, you need to know what the phosphate donor is, and what the amino donor is for each of these synthetic branches. Now the overall control of purine synthesis, 4 distinct points of regulation. Each of these is inhibited by downstream products. The synthesis of PRPP itself is inhibited by IMP, AMP, GMP, and by GDP and ADP also. These are the downstream products of this reaction. And the same is true for the synthesis of phosphoribosylamine, as we saw just a minute ago. Now the thing is, if you inhibit the synthesis of PRPP itself, you're also going to inhibit the synthesis of histidine and pyrimidine nucleotides. On the other hand, if you inhibit the synthesis of phosphoribosylamine, you're going to just limit the production of IMP, AMP, and GMP. Now, if we look at the branches going from IMP to AMP or to GMP, we'll notice that AMP feeds back to inhibit the synthesis of AMP, and GMP feeds back to inhibit its synthesis. Now, this offers a means of regulating this branch pathway so that high concentrations of AMP turn down the production of AMP and allow GMP to catch up. If we inhibit the top branch, the bottom branch will be used. Likewise, if we inhibit the bottom branch, the top branch will be used and will make more AMP. In this way, we get relatively equal amounts of AMP and GMP. And this is why we use different nucleoside triphosphates as phosphate sources in these syntheses. The production of AMP relies on GTP as a phosphate source, and the synthesis of GMP relies on ATP as a phosphoryl source. If this were reversed, we'd have big trouble. And so now we're going to talk about the salvage pathway. This is of prime importance for the synthesis of purines. The salvage pathway relies on phosphoribosal transferases, abbreviated PRT. These attach a preformed base that can be scavenged either from endogenous nucleotide breakdown or from the breakdown of dietary nucleotides. We scavenge the base and we add it to the activated ribose platform, PRPP. Now, the transferases are specific for the types of bases that they attach, and there are two major purine-based transferases. The first one transfers just adenine, so adenine plus PRPP yields adenosine monophosphate plus pyrophosphate, and of course we're going to hydrolyze the pyrophosphate to make 2 molecules of inorganic phosphate, and this is what's going to drive the reaction forward. Now, guanine and hypoxanthine bases are both transferred by the same enzyme, HGPRT, for hypoxanthine, guanine, phosphoribosal transferase. Guanine or hypoxanthine are added to PRPP to make GMP or IMP and pyrophosphate, and again these are driven forward by the hydrolysis of pyrophosphate to 2 molecules of inorganic phosphate, by which uracil and thymine bases are scavenged. Now, a defect in HGPRT leads to Lesch-Nyhan syndrome, and this is a pretty profound syndrome. It arises through X-linked recessive mutation. Individuals that have this have differing degrees of problems. Lesch-Nyhan syndrome leads to uncontrolled severe movement, also referred to as spasticity, severe mental deficiency, in some cases self-mutilation, including biting the fingers off or biting the lips or tongue, severe hyperaggression, and severe gout, and also kidney stones. A loss of HGPRT results in both an increase in serum PRPP and also an increase in serum urate, which is the catabolism product of purine bases. This increase in serum urate gives rise to gout and kidney stones. And because the salvage pathway is not working, there is an increase in the purine de novo synthesis. Deoxynucleotides are made from ribonucleoside diphosphates. We're going to reduce the 2' hydroxyl, and then we're going to phosphorylate the diphosphate to make a triphosphate. The exception to this is the production of TTP. This requires a folate derivative in a special pathway. For this, we need deoxyuridyl monophosphate, DUMP, and the cofactor N5 and 10 methylene tetrahydrofolate. This cofactor is a folate vitamin B9 derivative. This acts to transfer a methyl group to deoxyuridylate to make thymidylate. And so the products of this reaction are TMP and dihydrofolate. In the process of this reaction, N5 and 10 methylene tetrahydrofolate is converted to dihydrofolate. For this enzyme to become active again, we must reconstitute N5 and 10 methylene tetrahydrofolate. And to do this, we're going to use the enzyme dihydrofolate reductase. This is going to reduce dihydrofolate to tetrahydrofolate, and we're going to use the electrons from NADPH to do this. Once we have tetrahydrofolate, we're going to use a methyl group from serine to regenerate N5 and 10 methylene tetrahydrofolate. And so since this cycle is used in the production of TMP and not any of the ribonucleotides, if we target this cycle, we can shut down DNA synthesis. And so potent chemotherapeutics are used for this. Fluorouracil is a suicide inhibitor. It binds in the active site of thymidylate synthase and serves to shut it down by making a covalent bond to the enzyme that can't be broken. And so you can knock out thymidylate synthase. Other chemotherapeutics include aminopterin and methotrexate. These serve to inhibit dihydrofolate reductase because they look a lot like dihydrofolate, and so they can compete for the active site. And so aminopterin and methotrexate are competitive inhibitors of dihydrofolate reductase. Folic acid is necessary for the production of thymidylate. We take in folic acid in the diet. We cannot make folic acid. However, bacteria can make folic acid, and one of the things they use to make folic acid is PABA. This is p-aminobenzoate. In order to block folic acid production in bacteria, we can use sulfa drugs. These are antibiotics that look a lot like PABA. Sulfonamide is shown, and you can see the similarity in structure. Sulfonamide can act as a competitive inhibitor of the enzyme that makes folic acid. So by blocking folic acid production, we can block thymidylate production and thereby block replication in bacteria. The enzyme that catalyzes the reduction of the 2'-hydroxyl is ribonucleotide reductase. And we're not going to discuss this enzyme in great detail. It's sufficient to know that it has an active site and allosteric sites in each of the monomers that make up this dimer. This enzyme is primarily regulated through allostery. And one of the major ways in which this enzyme is regulated is through the actions of ATP to activate and DATP to deactivate. And so DATP feeds back to negatively regulate ribonucleotide reductase and decrease its activity, whereas ATP increases the activity of ribonucleotide reductase. The allosteric sites that are involved in specificity use differing amounts of these deoxyribonucleotides to fine-tune the activity of this enzyme. And again, this is to ensure that we do not have too much of one deoxyribonucleotide and not enough of the others. We want to make sure that we have relatively equal numbers of all 4 deoxynucleotides. This marks the end of Lecture 33. Have a great day.